Hello, good afternoon. Thank you very much for joining our virology conference. I am Claudia Florindo and I will be your host for this next presentation. I'm very happy to present to you Dr. Christof Hepp, who started his scientific work in Germany, in Cologne, in the biophysics group, where he studied the uptake of natural competent bacteria. Then he moved to Oxford and is now doing his postdoctoral research, working in the transcription, replication, and selective packing of influenza virus, and also SMRNA FISH-based RNA detection of virus. So today, Dr. Christof Hepp will be talking about developing single molecule fluorescence in situ hybridization for detection and structural biology of RNA virus. Welcome, Christof. Thank you very much for joining us. So okay, just a little information. Sorry, before we go on. This is, it's a live presentation. We will be very happy to answer your questions in the end of the talk. Please write on the presenter's chat the questions, and in the end of the talk, I will ask them to Dr. Christof. And now, without further delay, I'll send to Christof for your talk. Thank you very much for joining again, Christof.
Okay, thank you, Claudia. Okay. Yes, so today's talk will be basically about studying influenza replication and the selective packaging process with a method that is called DNA paint and is based on single molecule localization. So if you take a look at the life cycle of influenza, you have these ribonucleoproteins that basically contain the genetic information of influenza. So they are made up of the gene segments of influenza, which are negative sense single-stranded RNA. And this negative sense single-stranded RNA is then wound like around these nucleoproteins, which make up the structure of these RNPs. And basically, they start like it starts at the 5' and then basically goes down along the helix, turns around, and then just goes back to form this nucleoprotein helix. And on one end is thought to be the polymerase, which consists of, which is a heterotrimer of the 3 polymerase genes that you have in influenza. And essentially, when the influenza virus enters the cell and the RNPs get liberated by the acidification of the endosome, they end up in the nucleus, where they perform their 2 main tasks, which is basically replication, where they are making new RNPs, and also transcription, where essentially mRNA is made from the negative sense RNA of the vRNPs. Replication is a 2-step process because you essentially need to copy the RNA once to make cRNPs, which then are copied back again to arrive at the original vRNA. And a question has always been, what exactly is the mechanism of this transcription and replication procedures? How can transcription and replication happen with this defined structure? So very recently, there has been this new hypothesis on the influenza transcription mechanism which is called the processive helical track, and it's been derived by from cryo-EM structures of transcribing RNPs. And essentially this model is based on the assumption that there's a conservation of the RNP double helical structure throughout transcription, which basically can be seen here. So if this is a transcribing RNP, and this is the mRNA that is emanating from it, then you can in fact see that the structure seems to be more or less intact. Another thing that has been found by studies on the polymerases, also quite recently, is that the 3' and the 5' end stay bound to the polymerase during those transcription process. And if you take those two assumptions, what basically needs to happen for the for the ends to be bound while RNA is processed by the active center of that polymerase is essentially that the two strands of the helix need to move against each other. And an important question is basically, personally, I think this needs some verification other than the cryo-EM pictures. And also, more importantly, there's this question, does the same mechanism also exist for the replication steps? So one way to essentially address this problem or basically solve this question is that if like sequences could be specifically tagged with fluorophores and the distance of these fluorophores should change while the RNPs are actively transcribing. So basically, this would be the principle how you could prove this mechanism using fluorescence microscopy, and it has certain advantages over cryo-EM as well. For example, the cRNPs and the vRNPs can be distinguished, and you can hope to see whether there are like any mechanistic differences in these two steps of the replication process. And also another question would be like whether this assay can work on or like a prospect would be that like this assay could work on non-purified RNPs or even in situ in fixed cells. So you have a better chance of capturing these replication complexes, which have been very elusive. And also, of course, you would have a chance to see like these conformational changes within the natural environment of the cells, which would also be quite interesting. So the way to kind of solve this problem or to go about it is basically this super resolution protocol where we said, okay, let's do basically in situ hybridization of the RNP with a probe set of the but those probes, they won't carry fluorescent extensions like in fish, so that these RNPs can be seen directly. Instead, there will be short imaging strands that bind to these barcode extensions, and then essentially the transient interactions of the secondary to those primary probes produce this blinking pattern. And this blinking pattern can then be converted into super res images of the isolated RNPs by optical reconstruction microscopy. sorry. Oh, yeah. Okay, so yeah. So basically, as you can, I think I have to go back to the former slide. So as you can see, you get these like elongated accumulation of points that is about 100 nanometers, which is sort of the expected length of the RNP. Actually, in most cases, it is slightly shorter. And you can also analyze the length of those RNPs. So essentially, what I did here is I just fitted a covariance error ellipse to this cluster of localizations, and then essentially you arrive at the size distributions for the short and the long axis of the error ellipses, and then you can see that you actually have elongated RNPs. As you can see, like in the blue distribution for the long axis. So some but not all RNPs seem to be extended in this data set. Okay. I just jumped the slide. Okay. And here, what you could also do, essentially, is then, like, once we establish now that this works in principle, is measuring the distances between the different target sequences on an RNP. So essentially now we don't have probes that bind to the outline of this RNP, like all over the place, but only in specific places. So we have about 5 probes that bind very close to the 3' end, and we have 5 probes that bind at the middle of the segment, which would then be the end of this RNP structure. And if you do this with different imagers that correspond to the different barcodes used for those probes, you can sequentially interrogate the position of these primary probes. And basically then you can measure the lengths between sequences on an RNP. And you can compare this with a shorter distance that you choose just by changing the design of the probes, essentially. Okay. So essentially, what we then have is an overlay of 2 localization images of individual RNPs, and as you can already see, like kind of optically, it's like for the long distance, we usually have this usually the 2 localizations of the 2 ends of the RNP are spaced further apart than is the case for the short distance. And if you make a distribution for all of the RNPs in that data set, you can actually see that you can distinguish between those 2 distances. Although it has to be said that the long distance of 32 nanometers seems to be a little bit short at the moment. So this could be due to the preparation of the RNPs, or maybe also deviations of the RNP structure from what is expected from those averaged cryo-EM structures. Okay, another thing that we will use this approach for is the analysis of selective packaging of eRNPs into virus particles. This is a very interesting field, as it has been shown there needs to be some mechanism of selective packaging to explain while there are mostly 8 different segments in one virus particle, and there are usually not 2 copies of the same gene segment. So this needs to be a very highly selective process, as Sima already pointed out today. And one model how this selective packaging works is essentially the interaction of the RNA segments of those RNPs. So essentially you will get secondary structures that are thought to be quite similar to the base pairing that happens for double-stranded DNA. And essentially by cross-linking analysis, a lot of those localizations have been found that can form potential interactions with other gene segments. And I think what needs to be done is to take all of those interactions. Actually, there are more interactions that are shown on the picture below. These are just the most prominent ones. And yeah, basically, I think you need to kind of sort out which interactions are possible and which interactions are actually relevant to form or in this selective packaging process to form a virus particle. Yeah, so basically 2 things that we're aiming for is like the testing of the presence of all 8 segments at once in the same virus particle, which is something that hasn't been done yet. So there have been co-localization analyses of those segments, but I think it's been technically too demanding to do this for individual virus particles and then just interrogate all of the 8 segments at the same time. Another thing that will be important to figure out this process is essentially to observe the spatial configurations of those virus particles and then maybe get a clue whether they're like the segments inside the virus particle have all the same configuration, or if those configurations can in fact vary and you have like a set of configurations. And from this data, I think we should be able to identify basically RNA molecules or key RNA interactions for selective packaging. Also, if we contain this data, or if we combine this data with mutagenesis analysis. So this experiment is slightly different from the former one. First of all, we do not isolate the RNPs from the virus particles, but we immobilize them, fix them, permeabilize them, and then use the barcoded probes on those virus particles to cover all the segments that are inside this virus particle. There are secondary probes then, and basically the barcode of each segment codes for this segment. So essentially you have about 9 nucleotides, and all the extensions of the primary FISH probes have the same barcode. So what you do is you just add imager after imager and take it away afterwards, of course. And then you just read out all of the 8 segments until you can sort of get this spatial configuration of the segments in individual virus particles. Yes. And this is basically very new data where I had a first go at trying to make DNA paint images of this individual virus particles. So what you can see here is you usually have several localizations for each segment in these particles. And if you compare this with the scale bar and the actual size of a virus particle, which is about 100 nanometers across, you can see that all of these segments are within 100 nanometers. So that indicates that the virus particles are in fact complete and that they retain their shape. Moreover, you also can see that there is usually a spatial difference between the individual segments. So this actually makes me quite hopeful that we can analyze the spatial proximity of RNPs inside the virus particle and then deduce important information about packaging from this. Oops. Okay. So to conclude, the vRNPs can be resolved by DNA paint with barcoded primary probes and then short fluorescent secondary probes that serve as images in a DNA paint experiment. Moreover, the distance of the sequences on an ribonucleotide particle can be determined by targeting specific sequences. And that's actually, I think, very novel and valuable information that you can get there, because I think what has been done is people have identified the localization of the polymerase on those segments. And then essentially from cryo-EM pictures, you can also deduce some structural features. But I think I think it's very important to be aware that these RNP structures that have been that have been found by cryo-EM are statistical averages, and that actual RNPs might be very heterogeneous in their structure, also due to secondary structural elements that mediate the packaging. And last but not least, all gene segments in an influenza particle can be visualized by multiplexing. So yes, I really like to thank you for your attention. As I've already mentioned, I'm open to questions now. And yeah, just let me thank my group at the Biological Institute for Biological Physics at the Clarendon Laboratory in Oxford, Dr. Nicole Robb, which has now a group at the Warwick Medical School, and also the group of Professor Erwin Fodor at the Sir William Dunn School in Oxford.
Thank you very much, Christof, for your very interesting talk. We have a couple of questions, and I will start immediately with the first question. So, what is the spatial resolution that you get from this technique?
So the best spatial resolution that you can possibly get from DNA paint is actually very high. So you can, I think people have done 5 nanometers at the moment. And for my experiments, I would kind of expect like 10, 20 nanometers. Mm-hmm. But I'm just at the start of this, so I think this can be improved. Also, you have to keep in mind when we interrogate like one position on the RNP, we can basically repeat this measurement many times, right? Because we don't want to resolve a structure, we just want to determine where that target sequence is. And I think that can be done with a much higher precision. So this can be, I think there's no limit to the spatial resolution, but of course there's some systematic error in the measurements then. But it goes below 10 nanometers, I'm sure.
Yeah. So as for DNA paint, how hard is to get rid of the background noise in your preparations?
Yeah, so in my preparations, I think it's it's actually quite good, because as I've explained, I usually use virus preparations. So basically, all you have in there are virus particles, and I either lyse them or I don't. I lyse them for the RNP, so I don't for the virus particles. And then I think if something's really bright, it is usually the target RNA, because I have up to I have up to 48 different targets on an RNP. So that actually seems it's not that much of a problem. The interesting thing will be when you move on to infected cells, what will happen then? But actually, these experiments have been done in infected cells, so I think there is a chance that this will work as well.
Okay. And so we have a technical question here. It's how do you immobilize the virus particles for your staining?
Yes. So the virus particles at the moment are immobilized by evaporation. So essentially, what I like to do is I'd like to dilute them in saline 0.9%, and then essentially, I usually have a temperature around 40 degrees then, and I evaporate them so they're on the surface, and they seem to be quite stably on the surface if I do it this way. But there are alternatives, and if I see that maybe the structure of my virus particles is compromised, then I could also move to specific immobilizations, like using HA antibody interactions or other things like that. Okay. But at the moment, it's evaporation.
Thank you. So we have another question from Mark Gimira. He asks if have you seen particles that are packaged with fewer segments than 8, or are the packaged segments in different frequencies?
Yes, so this is a question I think I can't really answer at the moment. Because I've just I just started to do those experiments successfully, and it's a lot of data, and I still have to analyze the data, and I can't at the moment I can't really say what the chances are, but it seems if I do like a simple colocalization analysis of 2 fragments, then I think about 60-70% at least do co-localize. And that's basically what has been found in earlier publications where similar things have been done. From Peter Palise's lab, where they actually have done this, and I think they found about 90%, although they really purified their virus particles very well using a gradient. Yeah, so it's about 60-70%, I would say, of complete, or like not 60-70% of complete virus particles, but from there, I really have to do the analysis, so I can't really say how many. But the what I actually want to see is I want to see those incomplete virus particles, and then basically see how the presence of different RNPs depends on each other.
Thank you very much. So as for you have a barcoding system. So how many images can you get at the same time? And then how do you do the analysis afterwards? Yes.
So so usually in like one field of view, I will have about 500 to 1,000 RNPs, and this acquisition then takes about half an hour to an hour. And now I've forgotten the second part of the question. I'm sorry.
The second part was about the analysis. How do you do the analysis afterwards?
Okay. So for the analysis, we use Picasso, which is a special DNA paint program that was developed in Ralf Jungmann's lab at the LMU in Munich. It's basically just called, I guess, yeah, just Google Picasso and paint, I guess.
Yeah.
Or DNA paint. DNA paint, probably. Yeah.
So we have another question. Can RNA paint be used for live imaging to see RNA packaging during the virus particles assembly? And this relates to another question that was here and I haven't still asked. How compatible is this with the live imaging studies? So both questions.
I would say this is probably not a live imaging technique, because of course the imager strands need to get inside the cells somehow, and this is usually done with fixed and permeabilized cells. So I think the potential is to get this very good resolution, but for live imaging, I say I wouldn't see an application at the moment. So I guess something else has to be done for that, in order for that to work.
Okay. So you are using 8 colors.
Yes. I don't use. Yeah. Well. Okay. Just finish the question. Sorry.
Do you do the hybridization all at the same time, or you have a microfluidic system attached to your imaging device?
Yes. So okay, the different segments that I show as colors are actually not colors, but they are just they are basically imager strands with the same fluorophore, but this testing is done sequentially. In the future, I might want to use 2 colors just to have like a reference in case the virus particle moves. But usually what I do right now is I just exchange the solutions and wash, and I do this 8 times in a row. And a very large portion of the virus particles, I think about 95%, just stays attached to the surface. So that's actually something that works rather well. And it's also something that I had to kind of see before I went to start this project.
Yeah, that's actually impressive that you keep so many virus particles after those stripping and labeling again. Yeah. So I have another question here. What other possibilities are there to label RNPs?
So basically there are, I think, a couple of handles. What is, I think, very attractive and also very interesting if you want to, if you want to analyze the replication or the replication mechanism, is you actually would like to see where this polymerase molecule is. And this is also something that I'm looking into at the moment. So we actually have a lot of nanobodies for the polymerase, and there's actually there are very well described methods to tag those nanobodies with short oligos. So essentially you could also do this DNA paint and not just look for different sequences, but also look for the localization of the polymerase inside this complex. And then I think there's also for the nucleoprotein, if you want to have an outline or if you want to verify if what you're looking at is actually an RNP. I think you have tags for the NP proteins that are also compatible with super resolution imaging. So those are ways to kind of see those, basically analyze other constituents of these RNPs.
Thank you very much. And I have another question. Do you see this technique for analyzing the RNPs easily applicable to other viruses, as you do to your influenza?
Yeah, so this is a very interesting question. I think fish in general is actually quite widely applicable, and the hybridization with the primary probes should be as well. So actually, we're doing some experiments on coronaviruses and SARS-CoV-2 at the moment, and this essentially works almost exactly the same. And depending on how you prepare your virus particles, it actually also works very efficiently in a very short amount of time. So I would say yes, you can probably analyze most of those of the viruses. I guess if you have like a really rigid nucleocapsid that's made out of protein, that would potentially be a problem, because then you need a way for the probes to get inside. But if you have these quasi-crystalline nucleocapsids, then I guess there's just no way for the probes to go inside.
Thank you. I have another technical question here. So, how hard is to keep your RNAs, since many times the RNAs are a problem, and therefore it's hard to work with RNAs? So
Yeah, so basically when I do incubations for a longer amount of time, I usually use RNase inhibitor cocktails. I use RNase-free water, and then basically if you do all your solutions with this RNase-free water, you're usually fine. There's also PSA that is RNase-free, and so on and so forth. What I found is that actually if you so many of those FISH protocols, they work overnight, and I would actually say this overnight procedure seems to be a bit risky to me in that respect. So I think a couple of hours is usually totally fine with most FISH applications. And I think that also decreases the risk of just getting your RNA chewed up.
Okay. And on a follow-up of a previous question, so how long does it take you to image the overall setting? Because you image, you strip, and you image again. How long does it take you to do a full acquisition?
Yeah, so because the incubation with the secondary probes that have the fluorophores is not actually a stripping process, because the probes are very short, right? So they interact with the targets about half a second on average, and then they're gone. So when you remove the probes, you don't have to wait for a very long time at all. And if you add the new probes, then all the localizations that you should see comes from the new probes. And the procedure where I attach the primary probes that stay on the virus through the entire imaging process, that takes me about 3 to 4 hours basically. But the 3 to 4 hours are not the imaging part. The imaging part comes after that.
Okay. And do you need high power lasers to do the imaging?
Yes, so that that kind of depends on what I do. So it's also when you do this kind of experiment, it's always an interplay between the lasers and the camera. If you have a very good camera, you don't actually need laser powers that high. So yeah, basically what I'm working, or like the instrument I'm working with is nano imager from ONI. I can mention that here, right?
You can.
And basically I only use like, it's always a bit hard to calibrate, but I don't use the full laser power there. I use about 10 to 20% of the maximum laser power with that instrument. If you use too high laser powers, you get effects on your biological material, and you can actually deplete your targets. So basically, if your target sequences get destroyed, or if the virus particle gets destroyed during the imaging process, then of course you have to use either less laser power or some oxygen scavenging systems.
Thank you. And do you use chemical fixation in your virus particles?
Yes, so that's formaldehyde, basically.
Okay.
It's a very standard formaldehyde fixation. 4%.
And can I ask, now it's my question, how long do you fix it for?
Yeah, so I'm fixing for 10 minutes, and that seems to be enough for the virus, like isolated virus particles or RNPs, right? So I think that usually works quite well. There are some problems with formaldehyde fixation if you try this on whole cells. So it actually makes the efficiency of your fish staining go down, and you have to incubate for a longer time. That's, I think, how the overnight incubation periods come about that some people use in the protocols. Yeah, but this is basically, it's usually 10 minutes for those virus particles.
Okay, thank you very much for answering all these many questions that appeared. It was a very interesting talk. Yeah, I would stay for much longer talking to you about this. It's very, very nice. Thank you all the listeners for all the questions, and we will get back to any unanswered questions individually. And we really appreciate you being here. This talk will be recorded and it will be live on our Andor website. I have to thank you again so much, Christof, for this. And we will finish our talk today. And I hope I'll see you all soon. Thank you so much.
All right, bye.
Bye.